before that, let's have a look at these immunoglobulins that we have discussed. This immunoglobulin IgG, it's IgM. This is largest in size, okay. IgA is the second most abundant one, okay. And which one is carried by colostrum, if you remember? It is IgG. It is the most abundant one. This is the second most abundant one. This is the largest in size, and this is responsible for allergies. So this you have to keep in mind. Apart from this, all these antibodies, they are capable of producing a reaction against the antigens which come inside. The antigens could be bacterial antigens, or it could be a simple protein. See, antigen is going to be anything, any type of compound, any type of biomolecule which could bring about the formation of these immunoglobulins, which are of these many types inside the body. Now, these antibodies or the immunoglobulins are produced in response to specific antigens, and our body has a mechanism of memory, okay. Now, when we talk about innate immunity or the local defense system, we see that these local defense systems are aiming not in particular at a specific antigen. They are common for all, okay. Whatever response we talk about, they would give the same response whatever antigen is being encountered. But in the case of acquired immunity, it is a type of immune response which is specific, okay. Now, how it is specific? Because there is a characteristic feature of memory based response, and on basis of this memory based response, we have coined or we have been bestowed with vaccination thing, okay, all thanks to this particular property of the acquired immunity. So, an individual is not born with this immunity, first of all, because it is memory based. You don't have your memory when you're born. With time, you learn new things and that goes inside your memory. So, in the same way, the body is not going to be pre-programmed with all the data against the antigens. It has to learn from its period of growth that this is the specific antigen, then the memory would be created and that would cover under acquired immunity. Okay, I hope I am trying to make the point clear that because the innate immunity that the individual is born with is uniform for any kind of antigen, and acquired immunity is specific for the antigen, the antibody produced would be highly specific in response to the antigen and the quantity of antigen, of course. So, this has to be specific. When it has to be specific, it should have a memory that it could be able to distinguish between which antigen requires what response, and that would come with time. That is why it is known as acquired immunity, that an individual is going to acquire during lifetime. You must have heard that the infants are at high risk of certain infections. What is the reason? Because their body is not having this acquired immunity. Slowly and steadily, when they grow, they get many diseases timely, and though covered under certain categories, they acquire more and more data in their memory, and that becomes their acquired immunity, where specific antigen induces specific antibody production, or antibody production is otherwise known as immune response. Alright, this you have to remember. Now, in this acquired immunity, we have two types of immunity in action. One is the humoral immunity and the second one is cell mediated immunity. Now, in humoral immunity, what you have to remember is that there are special type of immunoglobulins. Those are known as B cells. Pardon me, those are not immunoglobulins. They are special type of lymphocytes. You remember the part of blood. One part of blood was lymphocytes. So, we have one type of lymphocytes known as B cells and other type of lymphocytes known as T cells, both of them being produced in the bone marrow and going to respective lymphoid organs for their maturation, are responsible for the acquired immunity. Alright, these B cells are the cells which are going to produce the antibodies or the immunoglobulins. Alright, in the acquired immunity, we see that the antigen brings about a response, memory based response, where the antibodies are produced. Now, who produces those antibodies? The B cells produce antibodies which are themselves produced in the bone marrow, and then they are transferred to different lymphoid organs like thymus or spleen for their maturation. These T cells, they move to thymus and they are matured over there, and these T cells are responsible for cell mediated immunity. Now, when we talk about cell mediated immunity and humoral immunity, what is the difference between these two? First point is, as I told you, the B cells are going to produce antibodies. So, in the body fluids, whatever kind of antigen is present, that would be encountered with the help of antibodies, and B cells would be responsible for that. But for telling that this is an antigen, there should be a mechanism. Because it is a memory based immunity, there is a mechanism which tells that this is self and this is non-self. So, who tells it? It is the cell mediated immunity which tells which part of the body is self and non-self. So, the T cells are responsible for distinguishing between self and non-self, that this is an antigen and this is the part of body. Otherwise, you can very well imagine that the antibodies have a strong tendency to eat up the body itself. That can also occur. So, there should be some mechanism which is responsible for carrying out that particular reaction or the process in a guided way, so that the self cells are not destructed, and that is because of cell mediated immunity. And when I say that this self and non-self, it has a wide application in the case of organ transplants. Pay attention over here, that what happens during organ transplant. You see, supposedly somebody is having a kidney transplant. Now, the kidney that would be transplanted in the body of the patient is a foreign object, huge organ itself, completely foreign to the body. Body is supposed to show an immune response to it. Alright, and this self and non-self immune response mediation, cell mediated response, has a huge role to play in the case of organ transplants. Now, when the organ would be transplanted, these T cells, they would come into function. They are going to make body aware that you have something that is foreign to the body. Now, at that place, B cells would arrive and produce antibodies and can lead to destruction of whatever transplant has been inserted into the body. Now, for that, we are going to see how many types of T cells are there. So, the first type of T cells are helper cells. The second type are killer cells. Everywhere you put helper T cells. Killer T cells. There are a type of killer cells otherwise also in the that kind of immunity, that is innate immunity, non-specific. That is why I have mentioned killer T cells. And the third one is suppressor T cells. Now, these three types of T cells are involved whenever there is a cell mediated immune response. Now, these three cells, what is their function? These cells, as you can see, they would be responsible for killing of the foreign particles. They would kill the cells which are present, and helper T cells are helpful in regulation of immune response. So, these helper T cells are going to regulate these two, and the suppressor cells, they are responsible for suppressing the action of killer T cells. So, these three cells are the types of T cells which are produced, and they work hand in hand so that the immune response is carried out, where the B cells are guided to produce specific antibodies. Now, supposedly this consider a human being who has encountered the disease for the first ever time. Now, the body would give all the possibilities, all the types of antibodies, and it is going to keep in mind that this particular disease was curtailed by specific antibody. So that is the primary response which under acquired immunity would be given. And once that response has been generated, a memory is created, and the second response, subsequent infection is met with a secondary highly attenuated response, and that becomes the principle of vaccination. Now in vaccination, you know very well that a strain that has been suppressed or a dead microbe is inserted into the body, it generates a primary response, and onto that the secondary response, if the infection comes, would be generated, and that becomes the principle of vaccination. And these things you have to remember, that in the acquired immunity we have two types of immunity, humoral and cell mediated. Cell mediated is governed by T cells, where T cells tell that this is self and non-self, very important aspect in organ transplant. Now the point is, supposedly this organ transplant is to be carried out, what would happen? The individual is to be provided with immunosuppressants. These immunosuppressants are going to target, firstly they are going to increase the activity of suppressor cells, and they are going to target the killer T cells. Okay. So the immunosuppressants are given to the individuals whosoever have undergone organ transplant, so that the cell mediated immunity is curtailed to a certain extent, and the individual survives that organ transplant, because the cell mediated immunity is responsible for differentiation of self and non-self inside the body. Once that differentiation has been carried out, now these T cells are going to give signals to B cells. Now how the signals would be sent? Certainly all of these are producing some protein intermediates. So it would be the protein intermediates being identified at the cell surface, which would be the site where identification and specificity would be considered, and immune response by a cascade of proteins involved would be carried out. So this is what you have to keep in mind when you study immune system.